Okay, so today I am going to cover genetic prediction and how to do Punnett squares. So you're welcome to pause this as you go through to be able to write any information down that you need to get that I'm going to go through pretty quick. So here we go. So Punnett squares are a tool used to create genetic probability, and probability is basically the chances of each offspring being able to get a particular trait from a particular set of parents. And so as we do Punnett squares, we're going to create boxes, and each of those boxes are going to be a percentage of the whole. So if we see a box with 4 squares, that's going to give us 25% per square, and that is the probability of that trait being seen in each offspring. So some vocabulary we need to go through is kind of define the difference between a character and a trait. A character is any inheritable feature, and a trait is the options of those particular characters. So for example, if your character is earlobes, the traits that you could have are either attached or lobed. So a gene, how this relates to genes, is the gene is the actual location of that DNA code for that particular character. Now, when we were figuring out these genetic traits way back when, we had no idea where these genes are located. Currently, that's what we're trying to find, is where is the gene for these particular traits so we can see the actual genetic code for them. And then the allele is the term used to describe what traits or genes that actual organism carries. So for each character, an organism will carry 2 alleles, and in the population you might see more than 2 alleles. For example, rose color has pink, red, white, and multiple colors. So there's multiple color of alleles in a population, but the actual one rose plant only has 2 of those. So when we use these alleles in genetic predictions, we are going to use letters to represent each of the alleles that a particular organism has. So we're going to go with a traditional format of assigning alleles. For each character, we're only going to use 1 letter. So every once in a while you'll see 2 letters being used per character, and that just gets really confusing. We are going to identify the uppercase of that particular letter as a dominant, and the lowercase is going to be the recessive. So an example here is if we have a character of P height and the trait is tall, we're going to use a big T, and if the trait is short, we're going to use a little t. If the particular trait we're looking at is not dominant nor recessive, so it's a complex trait, like a red and a white is going to give us pink, then we're going to use prime letters. So that indicates that these are not dominant recessive traits, but are more of a complex trait, and we'll get to those more as we move throughout our unit. So when we put together allele combinations, this is to identify what a particular organism has. So each of our individuals, again, has 2 alleles for each character, that one came from mom and one came from dad. This allele combination is called the genotype. So it's the actual genes that you have. What shows or is expressed, the gene expression of that particular genotype, is what we call the phenotype, so what we see. So if we want to see the color of hair that a particular dog has, what is the phenotype? We look at the dog. We don't have to go into the genes at all. And so our genotypes can, because we have 2 of each allele, they can be homozygous, which is also known as purebred. That means that the alleles are the same, or they can be heterozygous or hybrid, and that means the 2 alleles are different. So we would have 1 uppercase and 1 lowercase allele. So when we're doing our Punnett squares, we are going to follow a particular procedure, and with the monohybrids this seems a little redundant, but it for sure is going to help us as we get into our complex traits and our more complex Punnett squares. So the first thing you're going to do is list the character and assign the alleles for each of the letters. For example, the character is height. We're going to say T equals tall, little t equals short. You're actually going to write that out. Then you're going to identify the parental genotypes, and that's usually some type of story problem. So you're going to write out the cross, big T times little t. So this is a homozygous tall parent crossed with a short parent. And then you're going to identify the possible gametes. Again, in a monohybrid, this is pretty simple, but we need to write it out so we get used to doing that when we get to our dihybrid crosses. So in this case, you would have a big T or a big T from the one parent and a little t from the other parent. And then we're going to build a square to fit the number of gamete possibilities. And after we build our square, we'll fertilize and write in our offspring. So this square is a 4 square, and we're going to have the offspring here in red. Big T, little t are the genotypes of all the offspring. And then 6 and 7, we are going to identify the probability of each particular genotype, and they identify the probability of the phenotypes. So let's take a look at an example problem here. So in pea plants, we're looking at height, and tall is going to be dominant over short, and we're going to predict the offspring of a homozygous tall crossed with a homozygous short. So the first thing we need to do is to identify the character and assign allele letters. So we have height, tall is big T, short is little t. And then we're going to identify the genotypes of the parents, big T, crossed with little t, because this is homozygous tall with homozygous short. And actually, we don't have to tell you homozygous short, because for that trait to appear, both the alleles must be little. And then there's the possible gamete possibilities. The homozygous tall can throw only a big T. Homozygous short can only throw a little t. And so we can set up our gametes on the outside of our Punnett square. And this traditionally is a 4 square because it's a monohybrid, a one trait cross. However, you can reduce that to a single square because you only have one possible gamete from each parent. Then you're going to fill in your offspring, and from there you are going to then identify your genotype. So here is your genotype of your parent, which is 100% big T, little t, and the phenotype are all of those plants are going to be tall, because that's what the expression of big T, little t would be. Here's another example. Give a cross between two heterozygous. So again, identify your characters and alleles. You're going to identify your parents. Heterozygous means different alleles. And then the gamete possibilities. And here we can't reduce the size of our square because they're all different alleles. And so we put one parent on one side and one parent on the other. I guess, again, these are representing gametes. And then we fill in the box with the appropriate offspring lettering. Notice that we always put the dominant letter first if we see a heterozygous trait. Then we're going to identify genotypes, and here we have 25% big T, 50% big T, little t, and 25% little t. And the phenotype, 75% of those are going to be tall because they carry the big T dominant trait, and 25% are going to be short. So this is a situation where two tall plants gave a short plant because they were carrying the short gene. Alright, so in multiple characters, we are going to make sure that each character gets its own letter. So these would be for our dihybrid or 2-trait crosses. So for example of that, you're going to have height. Big T is tall, little t is short, and then flower color is our second character. And big P would be purple, little p would be white. Notice that I am doing the lowercase with a cursive p just to make sure that it's easier to identify. And then each of our gametes will have independent assortment for those particular alleles, and so they'll each receive one of each of those letters. So in this particular case, we have 2 characters, so we get 4 alleles, and we are going to FOIL as a kind of side point to be able to get the combination of alleles. So we have big T, big P, big T, little p, big P, little p as our possible gametes for a true heterozygous cross here. And then we are going to allow fertilization to take place, and during fertilization make sure that your letters stay in the same order, and if you have a dominant letter, that goes first. So for example, with this particular cross, you'd make sure that the T's go first always in the whole square, and that you would have the uppercase T first and the uppercase P first. And that just makes it consistent throughout the boxes so we can see those that are the same. Alright, so let's do a particular cross. In this one, we are going to be looking at seed coat and seed color. So the first thing we're going to identify are the characters and their alleles that they represent. And then our parents. So this is a hybrid for both traits, and one cross with homozygous round and green. Note here, green is recessive, so we don't have to say homozygous recessive. And then we identify our gamete possibilities for each of our parents, and we put those on the square, make a square that fits. So in this particular case, we only have a 4x1, and then we are going to go ahead and fertilize and cross. And here we have one of each genotype for 25% of each. And then our phenotype is round and yellow, so we have the dominant R with a dominant Y 50% of the time. And then we have round and green, a dominant R with two little y's 50% of the time. So hopefully that helps to be able to complete your Punnett Square activity. We'll see you soon. Bye.